So, as I explained in the last video, that sonic hedgehog specifies the identity of each digits according to the concentration of the sonic hedgehog in each of those digits. So, in one of the experiments done in mice, so they made a deletion of both, let's say, Glee3 or even sonic hedgehog, and in both cases, they found that the forelimb of the mouse is having this kind of phenotype. So we have extra digits. We don't have any more 5 only. We have much more. And these digits formed have no specific type. Okay. So what is giving the specification of this digit into 5, 4, 3, 2, or 1? It's the sonic hedgehog. I remind you that Glee3 is a transcriptional factor of the sonic hedgehog pathway. Okay, so you see that all the digits are the same, they look the same, we cannot differentiate between them. Okay, all are similar because we don't have sonic hedgehog. So now we'll talk about the regulation of the digit identity. Okay, who is giving the identity of the digits? And now we'll talk about another factor. Then sonic hedgehog will talk about BMP concentration. This BMP is usually present in the space between the digits called interdigital space. So here, for example, we are having a chicken hind limb, and here we have the digits. We have 4 digits in the chicken. In the first experiments, they tried to remove the first interdigital zone, the ID interdigital region number 2. It means this region. So after removing this interdigital zone present between the digit number 2 and 3, it turns out that the digit number 2 has been converted into digit number 1. Okay. And in the other experiment, when they removed the second interdigital zone, okay, the ID region 3, present between the digit number 3 and the digit number 4, they also found that the digit just before this interdigital zone, okay, it means the digit number 3 has been converted from the 3 into the identity of the digit number 2. Okay. So they tried to understand why, when we remove this interdigital zone, we have a conversion of the identity of the digit from one into another one. So for this, they just added, okay, beads containing BMP inhibitor. Okay, BMP inhibitor, because this interdigital zone express the BMP factor. Okay, they said that what would happen if we inhibit BMP in this zone. Okay, so they added beads that can express noggin, and noggin is one of the BMP inhibitors. Okay, so when they added the beads expressing noggin in this interdigital zone 2 between the digit number 2 and 3, okay, the digit number 2, okay, has been converted into digit number 1, exactly like what happened here when we removed the whole interdigital region. And in this experiment, when they removed this, or when they injected beads expressing noggin, an inhibitor of the BMP in this zone, the digit number 3 has been converted into digit number 2, exactly like what happened when we removed this whole interdigital zone 3. So this means that the BMP factor present in the interdigital zone is highly important for giving and for regulating the digit identity, since when we remove the zone, or when we inhibit BMP by noggin expression, we alter the identity, we can modify the identity of the digits. So now we'll talk about the third axis for limb growth. It's the dorsal ventral axis. This axis is controlled by the activity of the Wnt7a. So, according to the experiments, Wnt7a is expressed in the dorsal ectoderm. Okay, so Wnt7a is expressed in the dorsal ectoderm, but not in the ventral ectoderm. Okay, in the dorsal ectoderm of the chicken and mouse limb buds. So Wnt7a specifies the dorsal patterning of the limb. And they found that this Wnt7a expressed in the dorsal ectoderm will activate another gene called lim1 dorsally, and this lim1 will induce the expression of transcriptional factors, okay, essential for the specification of the dorsal cell fate in the limb. Okay, so Wnt7a is mainly important for the formation of the dorsal side of the limb. So in different experiments done, for example, they tried to make a mutant knockout mutation in mice for lim1. Okay, knockout mutation for lim1. They tried to remove lim1, and this produced a syndrome in which the dorsal limb phenotype is lacking. We don't have any dorsal formation or any dorsal side of limb that can be formed. And also loss of function mutation in human, okay, loss of function mutation for lim1 in human, since this gene is also conserved between mice and human. This produced a syndrome called nail patella syndrome. In this syndrome, the patella of the knee, okay, this patella bone is not normally formed. We have defect in this bone, but also in these persons, usually the dorsal sides of the limbs will be ventralized. Okay, we don't have any dorsal side formation, especially for the nail, where the location of the nail has been replaced by ventral structure. We don't have any right or normal nail formation. In addition, in mice which are knockout for Wnt7a, then they try to mutate the gene upstream of lim1, which is Wnt7a in mice. They obtained mice embryos having ventral foot pads, okay, on both surfaces of their paws. It means they cannot develop any dorsal side. Okay, both sides of the paws becomes ventral instead of having a dorsal side and another ventral side. You have to know that these three axes of limb growth coordinate together. So the dorsal ventral, the proximal distal, and the anterior posterior. So now I will talk about the coordination or the early interaction between the AER and the limb bud mesenchyme, okay, and the role or the specific interaction between AER and GPA. So as I explained before, FGF10 in the lateral plate mesoderm will induce the activation of the Wnt3a that will activate FGF8 in the ectoderm in order to form the AER. And this loop, FGF10, FGF8, will be maintained all the time to induce this kind of positive feedback loop will be maintained in order to form the proximal distal or to maintain the proximal distal growth of the limb. So here in the second figure, as the limb bud grows during development, sonic hedgehog, the main agent of the ZPA signaling center, will activate the expression of gremlin-1. Okay, gremlin-1, and this gremlin-1, it will prevent BMP from inhibiting FGF8 in the AER. It means this gremlin-1 will maintain all the time the expression of FGF8 in the AER in order to promote the growth according to the proximal distal axis. So you see that this ZPA, due to sonic hedgehog, will collaborate or will coordinate with the AER in order to coordinate the growth of the limbs during development.